Good evening. I'm Noemi Duran, and I would like to welcome you to the Faith and Science Fundamentals webinar series, organized through the European headquarters of the Geoscience Research Institute, where we have been exploring the relationship between the Bible, philosophy, and science regarding the origins. We are still in the biology module, and today we have our 11th session. Epigenetics and Pre-Adaptation. As you can guess from the title, today we will continue to talk about genetics. Genetics is the part of biology that studies heredity, and molecular genetics is the part of genetics that studies the molecules that contain the information that is passed from generation to generation. As you already know, that information is encoded in DNA and protected like a treasure in the nucleus of cells. We're going to start with a question. You know that DNA is an elongated molecule shaped like a spiral staircase. In the nucleus of a human cell, there are 23 pairs of chromosomes, which are 46 DNA molecules. Well, if we put the DNA of a single human cell in a row, one molecule after another, how long will it be? That's the question. And now I'm going to share a survey with you. Let's see here. There you have the question. And you have 4 possible answers. You have to choose the one that you think is correct. 8 nanometers, 3 millimeters, 1 centimeter, and 2 meters. In case someone doesn't know, a nanometer is 1,000,000 times smaller than a millimeter. Okay, you don't have to be afraid to answer. We're not going to fail anyone. Let's wait a little bit. We have about 60% of people responding who've already voted. Let's wait a few seconds more, see if anybody else decides to join in. 70% of all participants. Okay. I think that more or less everyone who wanted to participate has answered. We're going to close the survey. And it looks like there's a division of answers, but the majority have decided for the 2-meter option. We're closing this now. The majority, 73% of all who responded, said that 2 meters is the correct answer. Well, if you want to know the correct answer, you're right. The length of the DNA cell is approximately 2 meters. So inside of the length of the DNA of a human cell is approximately 2 meters. And you heard that right. Inside of each of your cells, there are 2 meters of DNA. How is that possible? Cells are microscopic. They can't be seen with the naked eye. And the nucleus is only a small part of the cell. How can 2 meters of DNA fit in there? Well, it turns out that DNA is a very long but very thin molecule that allows it to accommodate it or fit into a very small place if it's organized well. It's like this thread of spool, or the spool of thread, which is 100 meters long but takes up relatively little space because it's neatly wound around a plastic tube. And yeah, I'm sure you've already noticed that in the cell, everything is very well designed and that the same thing happens inside the nucleus. DNA has its own spool, made up of special proteins, of course, which are called histones. Histones are grouped by 8 by 8, into little bundles, and the DNA wraps around it to form a structure that looks like a string of pearls. Afterwards, the whole thing is compacted even more. The bundles of histones in DNA, called nucleosomes, spiral into thicker fibers, which fold even more. And in this picture here, we can see a model of all the folding levels. And although we represent DNA with the typical spiral staircase structure, the one that we see at the bottom part, it's actually not found that way in the cells, but rather wrapped in a very folded around histones, because if not, it wouldn't fit. When the cell is about to divide, the DNA becomes even more compact. DNA molecules get shorter and thicker and separate from each other. And at that time is when you can see the chromosomes with the microscope. These are the chromosomes. But normally, during the rest of the cell cycle, when the cell is not being divided, the DNA is in a state of intermediate compaction. And when we look at it under the microscope, we see a tangle of fibers or spots in which the different molecules are not distinguished. The fact that DNA is wound is a problem. We've already explained that DNA contains the information to make all the body's proteins and other molecules, such as RNAs, that the cell needs. That information has to be available to be copied and used when necessary. But what if the information that codes for a certain protein is in the center of the spool, covered by the rest of the structure, and is not accessible so that the proteins can find, read, and copy it? Well, as you can imagine, everything is already thought out. Most cells don't use all of their DNA. Remember that all cells in the body are formed from an initial cell, the zygote. So in each cell, all the instructions are there to make a complete organism. At the beginning of embryonic development, the cells are all the same, but soon they begin to specialize. Those that will be part of the nervous system acquire a star-like shape, very different from those that will end up forming the skin, liver, or muscles. But part of the specialization of cells is to block all the DNA that they no longer need. Neurons are never going to make digestive enzymes or muscle fibers, so the DNA that contains all that information is packed very tightly, and thus it leaves more room for the DNA that is going to be used on a regular basis. This has been known for a long time, but in recent decades, scientists have discovered the mechanism that the cell uses to block or unblock certain areas of DNA. And those discoveries are quite mind-boggling. Now we're getting into our topic of the day, epigenetics. The term epigenetics is formed by the word genetics and by the prefix epi, which means above or on the outside. If genetics studies the inside of DNA, the letters, genetics studies what is above or on the outside of DNA. Here we have a picture similar to the one we saw before about the levels of DNA packaging, but if you look closely, you'll see that in some places of the DNA and histones, there are small attached molecules of different colors. They're yellow colors here. These molecules are like labels that are placed above or on the outside of the DNA in specific places that affect the expression of genes. Some of these labels cause a previously inaccessible piece of DNA it was at the very back, to open and unwind so that the proteins that read and copy it are able to do so. And other labels do just the opposite. That piece of DNA is closed and blocked and cannot be read. This is very important because the information, the DNA letters, are still there in the correct order, but they are useless because they cannot be read. It's like if we had a book where 2 pages were glued together. The text inside is still there, but we can't read it or know what it says because we can't access it. The labels that open and close DNA are chemical molecules, usually methyl groups, like this one here. So when the labels are on, specialists say that the DNA is methylated. And these labels have two very important characteristics. The first is that labels are put on or taken off depending on environmental factors. What puts or removes labels from certain places in our DNA are things like food we eat or the amount of stress we are subjected to. The second is that the labels are inherited. They're passed to the generation that follows. When DNA is copied to make the sex cells that will be used in reproduction, not only DNA letters are copied, but also the labels on the outside. I don't know if you realize how crucial this is. Probably not, because it's really hard, it's difficult. And the scientists who discovered it also had a hard time realizing what they were seeing and the brutal implications that it has. I'm going to tell you about some of the investigations that opened their eyes to this. A research group was studying how rat mothers take care of their baby mice. They noticed that some rats are very affectionate and spend a lot of time licking and cuddling their young babies. But then there's other rats that are very scant and pay little attention to their young, and others have an intermediate behavior. Well, they studied the young of these rats until they became adults. And it turned out that the offspring of the unloving rats grew up to be aggressive, stressed, and abnormally behaved animals. Meanwhile, the rats that had received affection as babies were much healthier and calmer. To find out if the difference was genetic, that is, if the unloving rats had a bad mother genes that had been passed on to their children and therefore making them problematic because they had the same bad mother genes, the researchers decided to conduct an experiment by swapping the litters. What they did, they put the babies of an uncaring rat with a loving rat and to raise them, and then vice versa. They observed them once again until they were adults, and the result of the experiment showed that it didn't matter who the biological mother was. The important thing was how the young had been treated. Those who had been loved and cared for were still healthier and less aggressive than the others. regardless of their genes. When they studied the DNA of the two types of rats, they saw that there was a significant difference in the methylation pattern. So, the type of treatment of the mothers towards their offspring had changed the DNA labels of the little mice, and this affected the genes that were expressed in the adults. It made their behavior, or affecting their behavior and their health. There's another very interesting experiment that was done also with mice that had a mutation called agouti that makes the mouse yellow, obese, and have other health problems like diabetes. This mutation does affect the letters of the DNA sequence and is transmitted to their offspring. So the children of the agouti mice have the same appearance and the same problems as their parents. The researchers decided to feed pregnant mutant mothers, the yellow ones that were pregnant, a special diet rich in methyl groups. And it turned out that most of the babies were born with a normal appearance. They were brown, they were not obese or had diabetes, and they lived long and happy lives with no health problems. In this photo, you can see the mother and the son. When analyzing the DNA of the children, they discovered that the mutation was still there, but the area was methylated. It was labeled, just so that the gene was blocked and it couldn't be read, and therefore the disease did not manifest itself. Most of the studies have been, or more of the studies have been done with different species of animals, and the results are all in the same line. External circumstances, especially during embryonic development and in the first stages of life, but also after, cause changes in DNA methylation patterns. They place or remove labels, and this has consequences, good or bad, on the health of the individual as well as in their offspring. Although these types of experiments are done with people for ethical reasons, researchers have found that the same happens when studying the families of people who have suffered situations of great stress or great difficulties. For example, studies have been done on the descendants of people who suffered severe famines or who have been imprisoned in concentration camps or gone through wars. Some of the data that has been collected is logical and expected. The children of women who were pregnant when they went through these circumstances suffered the consequences and suffered from various health problems. But there are other observations that aren't as immediate. It turns out that the negative health effects continued later, not only in the children, but also in the grandchildren and great-grandchildren, even though they had been raised under normal circumstances and without depriving them of anything. And studying the DNA of these families has also uncovered abnormal methylation patterns. Gathering information from many studies, experts have concluded that external factors that affect DNA labeling have fairly predictable effects. So factors that we know are good, healthy, have positive effects, and factors that are bad that have negative effects. A healthy diet, doing sports, having friends, a good spiritual life. All of these things label DNA in such a way that the correct genes are opened and the genes that we don't want to be expressed are blocked. On the other hand, an unhealthy diet, drugs, stress, and things like that make certain genes that should be expressed unable to be so, and instead leave genes that should be blocked open. Cancer, for example, has genetic components. There are people who are predisposed to having cancer. It is written in their genes, and it seems that nothing can be done. But something actually can be done. Proper diet and a healthy lifestyle can help label those cancer facilitating genes so that they become blocked and quiet and they're not expressed. This doesn't mean that if we eat well, do sports, and have a good relationship with others and with God, that we are guaranteed of not suffering from cancer. You know that unfortunately it's not like that because I'm sure you all know someone who is very healthy that has gone through or is currently going through that terrible experience. But it's a question of probabilities. There are several scientific studies that link cancer to epigenetic factors and that give us hope. We can't decide the genes that we're born with, but we can control their performance in a certain way by placing the appropriate labels. I love talking about this because it's a reaffirmation of the health message that Adventists have been proclaiming for 2 centuries. Ellen G. White and the pioneers of Adventism knew nothing about epigenetics, but they recommended many of the things that current science has recently discovered. And let me tell you more. I'm going to take the risk that the theologians will scold me after for taking texts out of context, but there are some verses in the Bible that appear to speak about epigenetics. We can find them in Exodus and Deuteronomy, and they describe God as spreading the wickedness of the parents over the children to the 3rd or 4th generation. However, elsewhere in the Bible, we are assured that God does not make children pay for the sins of their parents. How should we understand this then? Well, perhaps God was warning that the actions of parents have consequences for their children, and that their children's children also. Consequences more serious and profound than anyone at that time could imagine. With regard to the topic at hand in these webinars series, the discoveries of epigenetics have enormous implications. If the DNA protein system was incontestable evidence of design, the extra layer of information contained in epigenetic factors makes the argument even more powerful. Epigenetics teaches us that living things are equipped with many more genes than they need to survive and reproduce. They contain a large amount of extra information that helps them adapt to new circumstances, turning certain genes on or off. The mutation-natural selection binomial cannot explain the appearance of epigenetic control because natural selection only acts on the genes that are expressed. In the words of Richard Dawkins in his book, The Blind Watchmaker, he says, evolution has no long-term goal. Natural selection has no purpose in mind. It has no mind and no mind's eye. It does not plan for the future. It has no vision, no foresight, no sight at all. But epigenetics tells us the opposite, that living organisms were built with foresight and that they are planned to be able to face future circumstances different from what they and their ancestors have lived until now, as well as what possible natural selection could cause. Also, the discoveries of epigenetics have affected the classical explanations of the theory of evolution. Do you remember when we talked about theoretically beneficial mutations that actually ruin things? Well, there's an example that appears in textbooks that refers to some blind fish that live in caves in Mexico. The traditional explanation for the presence of populations of these blind fish in caves is as follows. The ancestors of these fish lived outside and had functional eyes, but a group of them ended up living in the groundwater of a cave where no light ever entered. Coincidentally, because mutations occur randomly, one of these fish suffered a mutation that prevented it from making its eyes or creating its eyes. In other circumstances, this mutation would be very harmful, but inside a cave where there's no light, being blind is not a disadvantage compared to fish with eyes, because no one can see anything. You could even say that it's an advantage. The eyes, because the eyes are very complex organs, and the way they are made and maintained requires a lot of energy. So a blind fish could use all that energy for other things like foraging for food or reproducing. And so the mutation of blindness spread through the population until after X generations, all the fish were blind. It's a curious and interesting and plausible story, and it's also false. Because recently it has been discovered that blind fish or cave fish, they're blind not by a random mutation, but by an epigenetic mechanism. The genes for making eyes are present, but they've been inactivated by methylation. To show that this is the case, scientists have injected a substance that inhibits methylation into fish embryos, and those embryos have developed eyes. This is super important for the issues that we are dealing with here. It suggests that God put mechanisms in organisms that allowed them to adapt to different environmental conditions, such as living in the dark, and that the results of those adaptations can be viewed as bad. We may find it hard to imagine God designing blind fish, but it's a little bit easier to think that God gave the fish tools to survive after sin in less than ideal conditions. Like in caves where is little food and you need to save energy. So renouncing the eyes is one way to achieve this. It's a solution to having more energy, not wasting that energy. The discovery of epigenetics also provides us with a rapid adaptation mechanism. Organisms no longer depend on chance, on the coincidence that the rabbit that lives in the snow by chance suffers a mutation that turns it white. Instead, a dark-furred rabbit in a snowy terrain would detect the color change in the environment, causing the melanin-producing genes to be inactivated. And this makes it an adaptation intentional and much faster. There's a study that wasn't done with rabbits, but with lizards, and it shows exactly that. There is a type of lizard in the United States that lives in different substrates, and each population is the color of the background in which it lives. Some are lighter, others are darker, depending on the rocks where they live or where they normally live. The different populations have genetic differences between them, but the curious thing is that if a lizard is moved from its current environment, we take it from a lighter area and take it to a darker one, in less than a week it begins to change color to blend with the new environment. You can see some pictures here. It's actually the same lizard before and after being moved from a light desert environment to an area of dark volcanic rock. This means that the animal detects the change in color with its eyes, and that transmits some kind of message that causes a change in the production of skin pigments. As I was saying, creationist scientists are very excited about all this epigenetics, and we have several Adventist researchers working on this topic. I'm going to put an article in the additional materials so that you can read what is actually being done right now. But as always, there are things that we still don't know. We've said that epigenetic labels are passed down over several generations, but they don't last forever. For the adaptations to be permanent, the changes must pass from the external part of the DNA to the sequence of letters. And we still don't know how that happens, but we still continue to investigate. We're going to end here. Next week, we will finish our biology section with one of my favorite topics, one of the most beautiful topics that I like, which is natural selection and altruism.